and I'm currently advertising. And so this talk is a little slice of research that my lab does. I'm specifically interested in talking about non-local impacts in spatiotemporal models. And these non-local impacts, I think, occur a variety of ways. One of them is sort of behavioral plasticity or behavioral adaptation of individuals to their environment. And so, a species like this fox is going to respond to, its past history and its adaptive learning about prey resources and predators and other physical phenomena occurring across a whole landscape. And this behavioral plasticity sets up a dependence of every location on characteristics at other locations. That non-locality is, I believe, not thoroughly propagated through community modeling. And I'll make the case that empirical orthogonal functions is one way to get at that. So the general outline, I'll start with what I'm calling the grand habitat challenge. I'll give a brief review of empirical orthogonal functions, or EOF analysis. I'll talk briefly about EOF regression, and then application of EOF either for understanding single species demographic variability, sort of growth and mortality processes, as well as using EOF as an end-to-end ecosystem model for understanding climate impacts on marine ecosystems. So to start out with this grand habitat challenge, research is ongoing to generalize population models at a spatially downscaled level. So defining population dynamics or community dynamics at each location in each cell of a raster, and accounting for spatially correlated residual variation as a way of accounting for missing variables or otherwise some misspecification of the model. As sort of a toy model, a lot of times people do management using spatially implicit or spatially aggregated population dynamics models. And increasingly, those are state space models like Joanna has studied in her lab, in which, in a state space assessment model, there's biomass B at every time t, and that contributes to a change in biomass from one time year to the next. The delta B is the response, and you're trying to predict changes in biomass via a density dependence, a function representing density dependence given current biomass. There's some, harvest or otherwise a human impact of known magnitude, which is an exogenous driver. And there's also some environmental responses, some covariates X that could affect the prediction of production or change in biomass. And the simplest way that this is being extended to spatial population models is just doing that exact same calculation at each of capital S sites, S for space. So it ends up being essentially just an accounting exercise. With bigger computers, it's very easy to just define that same population model at each location. But that initial sort of extension has at least three kind of serious issues. One of them is stage structured effects. So we know demography varies with size, and size structure confers inertia and predictability. So people are building size structured extensions of that habitat model. There's non-local effects, including individual movement, or what I'll call environmental teleconnections. And there's mechanisms linking quote unquote independent variables that complicate attribution exercises or cause errors when predicting into new conditions. And so I'll spend this talk focusing on non-local effects. If you try to predict the impact of a characteristic on production at every other location, it leads to what I'll call an oversaturated statistical model. It could be non-identifiable theoretically, or it could be non-estimable given the amount of data we typically have. And my claim is that two feasible paths forward, one of them is to use movement models to identify dynamics that, individual dynamics. And the other is to use rank reduction methods, principal component analysis, or EOF, to extract spatial and temporal features that are predictive of correlations arising at geographically different locations. And so using the notation of my kind of previous slides, you could end up with this sort of oversaturated model where you try to predict catches and covariates affecting biomass at every location, where the potential set of interactions scales as the square of your scale. Or instead, you can try to do sort of rank reduction to extract features, or you can do a movement model that has kind of a low number of degrees of freedom to parameterize how biomass and density dependence plays out. So in the following, I'll be mainly using a set of R packages that are available online, and you can get to it at fishstats.org. For time, I'll kind of skip over that. So without further ado, the first case study is a paper that Lorenzo Cianelli and Mike Lizzo and I published recently in Progress in Oceanography. It's to define indices of ecosystem variability using biological samples of fish communities. And I like to tell this story. So this picture is a picture of Lake Alignagik in Alaska. It's a beautiful lake. There's a lot of work linking headwater processes, so the timing of melt, to lake ecosystem and environment. So, if there's a warm year in one headwater, that might melt the ice there. It causes cold water to cascade down into the lake. And so the variability in temperature in the headwaters is presumably highly variable based on geographically distant melting properties in groundwater. And the initial kind of like a box model of dynamics would suggest that all these headwaters mix in the lake, and then the outflow of the lake is more homogeneous. And so there's kind of this initial thought that fish, if they're trying to target a preferred temperature, might use the outflow as sort of a less variable temperature environment, and the headwaters might be a more variable temperature environment they could exploit to behaviorally modify their thermal environment. So a lab went out here and put temperature gauges, sensors in the headwaters and the outflow, and from the winter into the spring, their hypothesis was correct. But in the summer and into the fall, suddenly the outflow becomes highly variable at this predictable frequency of several days. And it took them a while to realize what was happening. So physically, the lake starts out cold and evenly mixed, and in the spring and summer, it warms at the top, and it sets up what's called a thermocline. And when the first storm comes through in the summer, it pushes all the water to one side of the lake, and sets up a tilt in the thermocline. And then when the storm passes, that thermocline rocks like the rocking in a bathtub for the rest of the summer. And when the thermocline gets above the surface of the water at the outflow, it dumps cold water out, and when it's above, it dumps warm water out. So the physical environment in this case sets up a frequency at which the thermocline will rock or sesh, and that is important to understand the ecology of this lake. So that's an example, that's kind of a classic example of what's called a teleconnection, where the physical environment creates a mechanism by which non-local conditions at the north and south of the lake, for instance, might be highly correlated due to the seshing of the thermocline, even though the middle is less affected. So so that, an ecological question is, how do you identify modes of variability from multivariate, noisy, zero-inflated biological data? It's called a dust bunny distribution. And the strategy is to use a multivariate spatial temporal model. So to kind of give a little bit more background, EOFs, or empirical orthogonal functions, is a way that oceanographers have for decades defined major modes of climate variability. So people might have heard of the Pacific Decadal Oscillation, or PDO. El Nino Southern Oscillation is defined similarly, and we're going into a La Nina affecting weather in my neck of the woods. So each of these oscillations is defined as a map representing how conditions differ from their average in a warm phase or cold phase, and then an index representing whether a given time is in a warm or cold phase. So it decomposes spatiotemporal variability into a map of responses and an index value. And people have previously linked PDO to fish returns for salmon, based on thoughts about thermal environment of the ocean. There's now a cottage industry of sort of trying to define like the second mode of variability in a different set of variables. So this is defined the NPGO, the North Pacific Gyre Oscillation, that's now thought to be determinate for salmon returns after a regime shift that occurred. So there's this cottage industry of trying to extract features from oceanographic data and relate it to time series of, in this case, salmon returns. So, the generalization of this kind of statistical multivariate generalization is to define a generalized linear mixed model in which you have a link function G, you have a set of intercepts for every category C, or species in this case, for multivariate data C, and time T at every location S. And on the right-hand side of this equation, there's the summation across NF factors. So there's an index map for one or more factors at every location S for every category. And then there's an index phase that's shared across categories. And then you define a response, a distribution for the response data that you have. And this could be optimized, as a likelihood model, a mixed effects model. So let's see, for sake of time, I'll skip sort of the machinery of this stochastic partial differential equation approximation to spatial random effects that is being used. And I'll start out by just trying to show people that this generalization actually does what it claims to do. So in the top left is the black line is the official, or the red, I guess, is the official PDO. And the PDO in a warm phase has that kind of lower temperatures than average in the kind of central North Pacific and elevated temperatures off the coast of Alaska. And the bottom left is the map of conditions using this statistical generalization of EOF fitted using VAST. Secondarily, the NPGO, the North Pacific Gyre Oscillation, is actually defined using sea surface height, which is correlated with sea surface temperature being fitted here. It's not actually calculated using the same variable, and so there's a lower correlation, but you capture some of the same variability. So next, if you do this, it's useful to define a statistical generalization because I can apply it to multivariate data, and I can use a Tweedy, a compound Poisson gamma, or a delta model, and fit it to zero inflated, right skewed, noisy multivariate data, in this case for 12 bottom associated fish and decapod crab species that have been sampled from 1982 through 2017 in this case. Using the version of this at the time, the first factor was, I didn't demean the data spatially, and so the first factor represents sort of the spatial niche or the long-term habitat selection. The second variable in this case is kind of a multi-decadal trend, and the third variable is this interannual variability, which explains a small portion of total variance, but explains a substantial fraction of interannual variability shown as a percentage in the parentheses. And this is an interesting case for reasons I'll get at, but and a point made by Dave Wharton and others, the benefit of doing community analysis or ordination of community dynamics using a model-based estimator is that you can take all your parts that you estimate and reconstruct what happened. It's hard to do that using NMDS or other multivariate techniques that people used previously for community analysis. And so in this case, I can use the parts of the model estimated to reconstruct dynamics for these three species shown here, where yellow is high density and blue is low density. And for people who know the system, this captures patterns that they know about. So the bottom row being arrow-tooth flounder that started out at low densities, mainly in the Southwest, and has increased in density and spilled onshore over time. In this case, Alaska pollock also had lower density in 2010 in the middle domain, shown in blue, and so on. So it's capturing kind of interannual variability we know about in the ecology of the system. The first factor represents persistent spatial variability. These are kind of reasonable maps of habitat selection for these species. The second one I'll skip again for sake of time, but the third one ends up being highly correlated with cold pool extent. So about 30 years of process research in this system thinks that dynamics are largely driven by the extent of cold waters in the Bering Sea. So in the winter, there's a polynya that's set up, an ice factory that's set up in St. Matthews Island. Ice is blown south. When it melts in the spring, it fresh cold water sinks to the bottom of the ocean and then itself spills southward. And that sets up a near freezing bottom pool of water that a lot of species can't physiologically cope with. And so they move out of that northern portion in cold years. And so in this case, only using biological data, I've reconstructed with a correlation of 0.87, sort of a known physical driver that was not included in the model at all. You also get this map of the species-specific response to cold temperatures. So in this case, Gadus calcogrammus or Alaska pollock, it's $1 billion industry. When cold pool is large or when this index has a positive phase, it has decreased density in the north and higher density in the outer domain. Similarly, Gadus macrocephalus or Pacific cod is avoiding the north and moving into kind of elevated, a few spots in those years. So, this is exciting because it shows that numerical abundance has this strong imprint of cold pool extent, and that biology can be reconstructed, or oceanography indices can be reconstructed purely from applying this generalization of EOF to biological data. So again, thanks to Lorenzo and Mike, and I'll keep going for sake of time. So the next piece is to think about how EOF could be used to optimize, make a map of conditions associated with a time series, like how to maximize the correlation between a spatially distributed mode of variability and a single time series. And so I'm going to do this for recruitment. So the stock recruit curve determines cohort strength and fishery potential for most fishes. And this occurs because juvenile stages or larvae are highly sensitive to climate impacts. There's this sort of match-mismatch theory where eggs have a little bit of oil, and depending on when the flexion, when the fish start feeding, if they are matched well with prey resources, they survive or don't. And so there's sort of an interest in matching stock recruit information, the production of juveniles, with spatially distributed physics. So again, the trick is going to be to define an EOF for physics. And so again, in the middle equation, I'm kind of changing notation from section to section, and I apologize. But there's again this sort of summation across NF factors, where each factor has a time series index, lambda, associating a factor with a given year. And then there's a map of conditions, in this case, epsilon, for every location X, for every category C associated with each factor. And then you can have, in this case, a identity link and a uniform distribution for physics measurements. And so the point is to take the dominant mode of variability from the physics submodel and include it as a covariate in a linear regression for some biological process. So pick your biological process. It could be salmon returns. It could be masting of maple trees to produce maple syrup. That being my stereotype of what Canadians might care about. I don't know, but the point being to take spatially distributed physics and link them up with sort of some spatially integrated biology time series. And in this case, I'm going to use a linearized Ricker model. So a Ricker model is sort of a conventional famous model for the stock recruit curve. And it's going to include this link to an EOF that's fitted to physics measurements. And so this is, again, kind of a cartoon of it, where the left-hand side is a submodel that explains interannual variability in some physical variable as a function of a time series and maps of response. And then that time series is also used as a covariate in a biological process. I should have said, I don't think I put in here that this was published in Global Change Biology this last year, and I'd be happy to provide references later. So in this case study, I'm going to take bottom temperature measurements from a summer bottom trawl survey, net sensors from 1982 to 2018. I'm going to take surface temperature measurements from a satellite over the same period, and then I'm going to use regional ocean modeling system predictions of bottom and surface temperatures to the end of the century. And I'm going to do what's called the delta change method to intercalibrate Earth systems model forecasts with my measurements. And then I'm going to link that with stock recruit records for pollock, cod, and yellowfin sole, the three biggest finfish fisheries in the Bering Sea. So if I do this without if I turn off this parameter linking the physics and biology submodel, I'm essentially just doing an EOF, an empirical orthogonal function, applied to physics measurements for bottom and surface temperature with Earth systems model forecasts of those same variables. And it's set up where they are loaded on the same index of variability and have the same response to it, but they have a different spatial map of initial conditions. And so the difference between those initial conditions is essentially what's called a delta change method or delta correction method to intercalibrate them. And looking at end of century forecasts, there's predicted to be bottom temperatures that are radically higher than anything we've seen by 2090. In the Bristol Bay and out to Nunavut Island and along Aleutian Islands. So next, if I turn on the parameter linking a mode of variability with stock recruit data for each species, I get a slightly different rotation of the physical feature that has a maximal correlation with stock recruit relationship. So I get a species-specific mode of variability that best explains recruitment, and I get the map of physical conditions associated with elevated recruitment for that species. So in this case, cod and pollock both do better than average recruitment when there's cold water in the southern domain. Cod specifically cues on environmental conditions resulting in cold water inshore, whereas pollock is more cued on physical conditions with cold water in the middle domain. And yellowfin sole kind of has the opposite map, where yellowfin sole seems to have elevated recruitment when there's warmer than average temperatures in the middle domain, shown in that left column, bottom left panel, with the red high temperatures in the middle. Finally, because this has ROM's, Earth systems model forecasts, I can do end of century forecasts of the stock recruit relationship for each of these species. So in this case, this is the left-hand panel is the estimated time series of log recruits per spawner. And the white is the historical period, gray is the forecast period every decade. And cod is predicted to have almost a 70% decline in expected recruitment. Pollock is expected to have a substantial decline as well. And yellowfin sole is predicted to increase slightly in abundance as oceans warm. So we can generate these end of century forecasts while intercalibrating physics and biology. So, I think the potential uses are to generate hypotheses about multi-decadal changes. In these cases, these are billion to $100 million fisheries. And so this is, a big deal to, the fishing industry. It also, generates these species-specific maps of environmental conditions associated with recruitment. And so that's useful to define places where if you were going to monitor physical conditions in a single location, that would be sort of the best location to monitor. And of course, it's useful to visualize and communicate these linkages between physics and biology. So this had a lot of co-authors that I'll list here briefly, and I'm happy to talk more about EOF regression. I don't have any follow-up research currently in the works. Two more studies that I'm going to pull out, and I'm going to keep going really quickly. So one of them is led by Arnaud Groos, who's now at NIWA in New Zealand, and a list of colleagues at the Alaska Fisheries Science Center, where we try to do this same EOF kind of approach to understand bottom-up drivers of demography for Alaska pollock. So for instance, this is showing the morphometric condition, or the relationship between body weight and length. And it's typically assumed to be an allometric relationship that's close to a cubic relationship. And if you take a log on both sides, you can see that you can end up doing a model for alpha, and alpha is called Lacren's condition factor. So alpha is essentially a measure of how fat a fish is relative to what you'd expect for its length. Similarly, you can fit a spatiotemporal model to other data that represent demographic processes. So growth is reflected in size, the fork length, the mouth to the caudal fin length, or other length measures for the age of maturity. You can fit a univariate spatiotemporal model to that. You can fit a multivariate model to the prey biomass per predator biomass. We have a paper in Fish and Fisheries on this to get at their, pollock's consumption of prey species, euphausids and copepods and shrimps. Or you can do what we saw already and do movement using numerical abundance. And so I'm going to call that initial factor model an exploratory factor analysis. And then you can also think about sort of confirmatory factor analysis, in which, in this case, we're going to pre-specify a mode of variability using the cold pool extent and see if that cold pool extent is explaining the same thing as the mode of variability. So if we do this, we can get a spatial map omega associated with kind of average conditions for each of these processes. And some numerical density is highest in that middle domain, like we've seen repeatedly. Morphometric condition is highest in red on the southern area, where they're about 10% heavier for a given length. The length at age 7 is highest inshore and lower offshore. Copepod biomass, the final 3 are different prey species consumption. And so copepods, small crustaceans, are eaten most in the outer domain. Euphausids or krill are eaten most in the south. And Mysidaceae, or shrimps, are eaten most in the inner domain, kind of again in the Bristol Bay and Nunivak Island. So those are kind of the average spatial expressions of those demographic processes. But in each of these, fitting each one individually with an EOF, we get a time series shown in gray on the left-hand side, and then I'm lining it up with cold pool extent shown in blue. The correlation between the dominant mode of variability and cold pool is 0.6 in this case, only fitting the pollock. It's lower for a single species because there's more noise for that individual species than all species combined, like we saw before. For condition, the correlation is 0.56. For length at age, it's 0.35. And we'll see stomachs in a second. The middle column is the spatial map associated with a positive or negative phase of that mode of variability. Then the right-hand side is what we're calling the confirmatory factor analysis. It's a spatiotemporal model with a spatially varying response to cold pool extent. And in general, those maps also are correlated with the maps of the mode of variability, with the exception of condition. So, and then this is for stomach contents. Stomach contents has a 0.66 correlation when doing a multivariate model of stomach contents, showing that for instance, in warm years, there's higher consumption of copepods in the kind of outer domain, higher euphausid consumption in the south, and so on. So I mean, I guess the conclusion is that we can do this proof of concept showing that we can get this sort of demographic fingerprint of bottom-up oceanography on, well, on habitat utilization. For pollock, in this case, all of these demographic processes have their dominant expression of variability that's, that's correlated with cold pool extent. So there's some physical process that's correlated with cold pool that is also presumably driving these these demographic processes. And this confirms that there's a strong bottom-up driver for this billion-dollar industry. As climate change eliminates sea ice, for instance, summertime Arctic sea ice by 2030, as currently projected, it's going to cause a huge change in all of these demographic processes for Alaska pollock. And then finally, for maybe 5 or 10 minutes, so we have some time for questions, but acknowledging that we started a little bit late, and sorry for that. The final application is for kind of whole of ecosystem modeling. So what happens if you try to assimilate ocean physics, primary producers, secondary producers, juvenile fish, adult fish, seabirds, fishing effort, all in the same model? Typically when we do community analysis, we just do it for individual trophic levels. Like classic meta-community theory is for, top consumers or, plant communities. And I don't think that we've gotten into kind of meta ecosystem modeling using spatiotemporal models very, very well. So in this case, this is a record of the different variables we're fitting. Each of them has a different set of years that it's available. I'm including a 10-year forecast through 2030 for the physics. Some of them are spring or fall variables. Some of them are vertically integrated or bottom or surface, and EOF is able to stitch together these sort of different sampling processes. Let's see, so conceptually it kind of looks like this, where we've got, wintertime satellite temperatures, we've got summertime bottom trawls, we've got spring and fall primary and secondary producers, we've got a spring and fall fishery, and not shown here, we've got seabirds in the fall. If you look across all these years, it's on nearly 150,000 sampling occasions, and different data sets pop in and out. So again, sort of spatially, seasonally unbalanced sampling data for this ecosystem, which I'm sure is typical for all ecosystem sampling programs. Okay, so that again, I mean, this structure I typically use is to have sort of this spatial map of habitat utilization. So in this case, we've got a map of bottom temperature and surface temperature at the top. We've got the delta change method intercalibrated bottom temperature and for a forecast, a ROMS forecast. We've got chlorophyll A in the second row. We've got some calanus or copepods in spring and fall. We've got juvenile age 0 pollock. And then we've got 5 different fish and crab species. And then the bottom row is the catcher processor fleet, a fishery in the spring or fall, and northern fulmar and shearwaters or birds. So there's a whole story about the ecosystem function here that I don't have time to talk about, but just to pull a few pieces out, you can see a bit of what's called the spring large copepod chlorophyll in the second row right column. That's what's called the Bering Sea Greenbelt near Jamchug Canyon. So it's thought there's the biggest ocean canyon in the world cuts from the slope up onto the Bering Sea shelf, and in the fall, dominant water currents are flushing nutrients up to the surface and causing this amazing chlorophyll bloom there that then drives deep water foraging of fishes, that when they die, they float, and that attracts albatross. There's amazing ecosystem stories that are kind of set up by some of the physics shown here. Again, pollock has this sort of characteristic distribution. Juvenile pollock are more in the south. The spring capture processor fleet is mainly in the south, and in the fall it moves northward, and so on. So the main point of this is that the dominant mode of variability, surprise, surprise, is again highly correlated with cold pool extent. So in this case, we can do again, all of these collectively have 80% correlation of the dominant mode of variability with this physics measurement of cold pool extent. And fitting all these variables simultaneously allows us to do a decadal forecast. Our current forecast is that, this is an ensemble forecast, that recent warming conditions are likely to persist, and under these conditions, we'll have a low cold pool. Let's see. So, and each of these variables has a map of condition changes in their spatial expression under smaller cold pool. So, again, the spring Bering Sea greenbelt has lower intensity when under warming conditions. A lot of the fish move north, and also the seabirds are impacted. And so given that forecast, you can do these, end of century forecasts for each ecosystem component, or more interestingly, you can actually map out sort of the overlap between ecosystem components. So Schoner's D is a measure of overlap between system components. And under future warming, we're predicting that arrowtooth pollock and arrowtooth flounder and adult pollock have increased overlap. Adult pollock and juvenile pollock have increased overlap. And so a lot of the predator-prey interactions are intensified under future warming, but that simultaneously there's lower than average overlap with the adult fishery under future warming. So again, I mean, I guess that, not to be too dire about it, but I think that, anybody who is earlier in their career is going to face, climate impacts in their personal life and in their children's lives. And I think that, we all face this grand challenge of figuring out how climates can impact things. And, one phrase is that the Arctic, what happens in the Arctic doesn't stay in the Arctic. So there's Arctic amplification that's going to cause changes in weather patterns, sparking wildfires in California and causing me in Seattle to be exposed to harmful levels of smoke. These are kind of classic teleconnections where, physical drivers can drive ecology and human outcomes half a world away. I think climate is sort of the ultimate example of ecological teleconnections. And we need to get smarter as ecologists and statisticians about how to build in non-local interactions into our ecological management. So again, emphasizing, well, I forgot to revise this. I'm calling it 3 instead of 4 challenges now, but I'd love work from anybody there to think about size, stake structured effects, non-local effects, and attribution when there's mechanistic associations between covariates in building spatial population models. In terms of postdocs, I can actually circulate an announcement for the first one, and hopefully I'll do that after the talk. I've got a little 1-pager, but we've got funding for a 2 to 3 year postdoc on climate impacts on zooplankton in the Bering Sea. So you saw us talk a bit about that. We've got a 30-year time series for zooplankton, and it's one of the richest taxonomically and seasonally in the world. And we're trying to intercalibrate that with Earth systems models. We've seen a little bit of that shown here, but I didn't show any of our climate forecasts of zooplankton. We've got a regional ocean model with 6 different categories, and we can validate that model and also do a statistical forecast. And then the second one is currently a 1-year postdoc, although I anticipate getting more funding, to do a data integrated movement model, combining sort of Lagrangian tracking for conventional tags, archival pop-up tags, satellite tags, and acoustic tags, combining that with point count or kind of Eulerian accounting of like survey data, transect or random or fixation point count data. So feel free to send me an email about either of those topics. Both of them, I think we'd be happy to wait for a good candidate. And both of them, we're looking for a statistician who's interested in ecological questions, but doesn't necessarily have a strong ecological background. We just need somebody who's technically competent, and we can go from there. So email me about either one. And finally, I'll put up, I don't think this is the full list of people, co-authors throughout the paper, but thanks to them. And hopefully I have a few minutes for questions. Thanks.